MSc, PhD in Ecophysiology, and B.Ed. M.Ed, and MCA and MA English. Former I was lecturer in botany. Then I became principal of a college in Mathura. Then I retired as a principal. Now I am teaching to the civil services and 12th and UPCSC board. Today I am teaching immunity. Immunity is a very important question and it is asked in the examination. The fungi and certain pathogen produce antigens and the organism has the power to fight. It is called immunity and study is called immunology. First of all, non-specific immunity, physical barrier by skin and mucous membrane, physiological barrier by lysosome from enzyme, and gastric justification by chemicals and destroy pathogens. Cellular barrier by polymorphic fungus forms and neutrophil are present, and monocytes form natural killers. Kills to the phagocyte cells when amoeba pseudopodia kill weak, and it is called immunity, and neutrophil also form monocytes. Macrophage of skin form monocytes, and liver Kupffer cells form phagocytes. Phagocytes, if you want to see the phagocytes, you can see the diagram of phagocyte. First phagocyte, then it form phagosome, then it form a structure called pseudopodium, and the cycle is completed and immunity is occurred. Next, cytokinin barrier. Cytokinin barrier is produced by natural nucleic acid, viral nucleic acid, then it form DNA and new virus are produced. These produce interferon. Interferon kills to the DNA and immunity is maintained. Now we see the cell mediated immunity. Cell mediated immunity forms by antibody, and in this first plasma cell, then beta cell, then क्या है ना नहीं क्या है
Antigen.
then antigen cells,
Okay.
then helper cells, then memory T cells, then all these memory T cells and plasma cells form antigens, and plasma cells form memory phytotoxic cells, then they form active cells. They are non-toxic and they form the cytokinin. Then cytokinin to the helper cell, then they form antigen nursing cell, then they form interferon, and then they form बचलिए क्या है
Stimulated.
simulated क्या है
Simulated, yeah.
simulated cells.
Gives cells.
Are called T cells, and they form antibodies. They antibodies form antigens. Antigens are special protein. If you see a protein, then they produce nucleoprotein, lipoprotein, glycoprotein, and polysaccharides. Antibodies are also a special type of protein, and they produce immunoglobulin, and they contain 2 light bands and 2 dark bands. If you want to see the structure of the antibodies, then we see heavy chain, then we see the light chain, then we see the antigen binding site on the both sides, If you see the diagram of this, then we are looking this. They are very important, and this, that is antibody structure. Immunoglobulins is of two types. First is IgA, IgB, and IgM. And Ig antibodies near about 80%. They are found in placenta, and they leave to the blood and reach to the serum. IgA produce anti-allergic things first. Antibodies help in agglutination, help in neutralization, help in precipitation, help in openization, and then it forms complement enzyme cell mediated cells. If you see the antibodies, first percentage of T cell 10%, then 1.3% IgD, then Ig 0.05%. IgA help in, because it is found in colostrum, and it is found in milk of animals. Then in small amount it is found in lymph cells. Then it is found in even neutrophils. Then it is function. The mucous membrane contains these cells, and lymph cells are also found in this, and mast cells are also found in this. Khatam.